Thank you very much for joining webinar today. I'm Mayumi. I'm currently working as a researcher at Ajinomoto at the Institute of Biofine Institute. So yeah, today the title of my talk is CD-AOF StemFit culture systems and their applications with human iPS cells technology. So before starting, we have been working with the stem cell scientists for years globally and found that it is still difficult to maintain stable and reproducible stem cell culture at the lab scale. So while culture materials themselves are one of the key factors, we at Ajinomoto have been working to establish better culture systems to solve the potential challenges faced by stem cell scientists. So today I'd like to address these challenges by introducing our latest product developments. So firstly, I'll provide an overview of our company, Ajinomoto. So our headquarters is based in Tokyo, Japan, but we have 35 business sites globally. And our core technology is the manufacturing of amino acids. And also based on this amino acids research, we have built a diverse business groups encompassing products such as seasonings, frozen foods, cosmetics, electronic materials, and pharmaceuticals. And our development of, sorry, like the trouble is happening right now. Okay. So our development of cell culture media, StemFit, also started with the amino acid technology right here. So our first human iPS cells prototype culture media, StemFit AK01, was developed in the collaboration between Ajinomoto and the Center for iPS Cell Research and application at Kyoto University, CIRA. At the time, CIRA needed a better culture medium for human iPS cells, which enabled single cell passage that would deliver high quality and consistent performance. As amino acids are one of the essential ingredients for culturing cells, Ajinomoto's research expertise in amino acid nutrition and analytical techniques and know-how in biotechnology were instrumental in developing an improved cell culture media. And excitingly, in 2014, CIRA's iPS cell establishment and maintenance expertise and evaluation techniques were collaborated with Ajinomoto's amino acid technology to successfully develop the world's first single-cell passage optimized media for human iPS cells. So before introducing the StemFit culture system, I'd like to briefly clarify the definition of animal origin-free grade products. They are primarily three graded categories assigned according to the components and the medium. Firstly, serum-free grade products don't contain serum. However, they do contain other animal-derived products such as BSA. And in this grade, the components are undefined and large lot-to-lot variations can be found. Xeno-free grade here has the same properties as the serum-free grade, but with the additional removal of animal-derived products. However, human-derived components such as human seroalbumin or HPL are still present in xeno-free media. Because of this, there is a risk of immunogenic viral infection or lot-to-lot variations due to donor differences. Lastly, animal origin-free grade further removes human-derived products. Therefore, the risk of immunogenic contamination is negligible and lot-to-lot variations are minimal. In addition, in chemically defined graded medium, all of the chemical structures of the components are identified. As I mentioned in the previous slide, animal or human-derived products have many risks in clinical use. Therefore, by choosing to use chemically defined animal origin-free culture system, CD-AOF products, you are not only improving consistency and robustness, but also ensuring that the transition from research to clinical applications will be easier and less likely to encounter major setbacks. CD-AOF culture systems provided by StemFit from expansion to differentiation of hPSCs will provide consistent performance and ease regulatory compliance, both of which are essential for clinical use. So I know many of you already use StemFit and are aware of its core benefits, but I'd still like to briefly summarize its advantages here. Superior and stable growth, consistent performance, and high single-cell cloning efficiency, less label-intensive, cost-effective culture, and regulatory compliance. So the first advantage of StemFit is its superior and stable growth in a feeder-free culture system. It easily supports transition of on-feeder cells to feeder-free cultures through a simple protocol, as shown here in the figure. Additionally, we have confirmed that single-cell passage with StemFit is compatible with different, like many matrices here, such as iMatrix 511, vitronectin, and laminin 521. So this graph shows the fold expansion of cells cultures with StemFit at varying cell densities. In both densities and on different matrices, efficient cell growth was confirmed. As you can see in the graph, usually fold expansion of cells is more than 100 times. Next here show the consistent culture performance of StemFit. This evaluation was carried out at the Cell and Gene Therapy Catapult in the UK. In this evaluation, StemFit AK03N or Basic 03 was compared with other expansion media for iPS cells in terms of population doubling and gene expression profiles. So as you can see, StemFit media showed the highest growth rate, and the cross-link figure here of gene expression profile also showed that cells cultured with StemFit have genomic stability through 5 consecutive passages. And karyotyping analysis also showed that cells could be cultured without any karyotype issues through 10 passages. These results confirm that robust and reproducible and consistent cell expansion is possible when utilizing the StemFit culture system. The StemFit culture system is also able to support the single-cell cloning. Here we cultured the cells for 3 passages using 3 different culture media respectively, followed by a cell cloning experiment, utilizing the serial dilution method. This was conducted on 2 different cell lines. In both cell lines, StemFit showed the highest cloning efficiency due to superior growth and high colony forming efficiency. Therefore, we recommend StemFit not only just for expansion, but also for the establishment of high quality cell lines. We also provide less labor-intensive cloning protocols when using StemFit systems. So in the conventional method, usually the plates are pre-coated with matrices such as iMatrix 511 prior to cell seeding. To reduce the time and labor prior to cell seeding and cloning, we suggest utilizing the mix method or uncut method for the seeding process. In the mix method, extracellular matrices are mixed with the cell suspension so that cells are seeded together with ECMs. The cloning efficiency between the mix method and the procut method were equivalent, and in both procedures, the expression of pluripotent markers, OCT34, SSAF4, were highly positive in both conditions. So we hope this method can be utilized to reduce your label during cell cloning. This slide shows the cost-effective and low labor cell culture protocol developed for StemFit. In the conventional protocol, 2 ml of medium are used per well for a 6-well plate, and the frequency of the medium change is 6 times per week. In the StemFit protocol that we developed, the amount of the medium per well is only 1.5 ml for a 6-well plate, and the frequency of medium change is reduced to 3 to 4 times per week. Therefore, by using StemFit, the total medium consumption and the cost will be 50 to 60% lower than in the conventional protocol. Here we show a few scheduling examples of medium changes in passaging during the week. So in these schedules, regardless of passage timing, like Tuesday or Wednesday or Thursday, you can skip the weekend medium changes for good. Taken together, the reduced labor and cost enable StemFit to provide a unique and highly effective culture system. So StemFit has regulatory compliance for clinical therapy and product development. Japanese Pharmaceuticals and Medical Devices Agency, PMDA, confirmed that our CD-AOF mediums are able in clinical applications. We have also launched a SHE GMP grade media, Basic 03 GMP, to meet the US regulatory requirements. Several clinical researches and cell manufacturing process constructions are proceeding with StemFit. For example, the world's first clinical study with iPS cell-derived corneal cells is already underway utilizing StemFit. As a culmination of all its advantages and regulatory compliance, StemFit has become the standard clinical grade medium for human iPS cells in Japan. As I mentioned in previous slide, several clinical research studies have proceeded with StemFit, such as studies on Parkinson's disease, spinal cord injury, and corneal transplantation, and heart failure. In addition, number of the publications utilizing StemFit have been increasing year by year, and including publications in significant scientific journals. Ultimately, we hope and believe that StemFit will help translate as much early stage regenerative clinical studies, regenerative medicine research as possible into clinical study by providing high quality CD-AOF STEM systems that support all stages of research. This is the StemFit product family. So Basic 02 was our original commercial offering, and Basic 03 or 03 GMP was developed to be CD-AOF and gained PMDA or FDA approval. Basic 04 or Basic Complete Type followed as our next generation media with all the same benefits at one new low price. With Basic 04 CT, Complete Type, you can upgrade your culture system to CD-AOF at no additional cost compared to Basic 02. So far, I have introduced the advantages of the StemFit for human iPS cells expansion mediums. So from this section, I'd like to introduce our newly launched product, StemFit for Differentiation, also referred to as AES401. In the scope of regenerative medicine research, the cell culture process generally consists of two principal phases. The first phase is the generation and expansion of iPS cells. In this stage, the expansion medium series I shared in the previous slides can be applied. The next stage is differentiation. StemFit for Differentiation, or AS401, can support this stage as a CD-AOF supplement that induces cells into the desired lineage. In the bigger picture of translating basic research into clinical research, and even further into cell manufacturing, it is important to utilize the CD-AOF culture system for both the expansion and differentiation of cells. Additionally, the earlier on that our CD-AOF system is employed, the smoother the translation from basic research to clinical to total cell production should be, resulting in accelerated research and product development. In iPS cells differentiation, usually animal-derived supplements such as FBS or serum substitutes such as B27, KSR are often used. And AS401 is able to substitute these at CD-AOF grade. By utilizing CD-AOF supplement for cell differentiation, performance of differentiation will become robust and consistent because of negligible lot-to-lot variation, and further clinical application is possible. This figure here shows some examples of applications for AS401 as a supplement for cell differentiation. AS401 can be added to a basal medium, such as DMEM or RPMI, at 20% of total volume to make a basic differentiation medium. Subsequently, differentiation factors such as cytokines or small molecules can be added to this basic medium according to the necessity and the objective lineage differentiation goals. In today's talk, I'd like to share 3 examples of lineage-specific differentiation utilizing AS401. This table showed some examples of lineage-specific differentiation in which AS401 was able to replace serum or serum substitutes, which are shown in red circles. So YCU here stands for Yokohama City University in Japan. Firstly, AS401 was developed in collaboration with YCU as a CD-AOF differentiation supplement for hepatocyte differentiation, and now preparing for clinical study with AS401. In next few slides, I will share differentiation examples of hepatocyte and dopaminergic neuron and hematopoietic progenitor cells. Before sharing our lineage-specific differentiation experiments, I'll show the process of how researchers had Ajinomoto build CD-AOF differentiation protocols. There are typically 3 developmental steps. First, we thoroughly review papers related to our target cell type. Generally, we select protocols to test by evaluating their robustness. We investigate whether the protocol has been tested by different cell lines or whether they use the animal-derived components for the culture. And to determine the reliability of the papers or the protocol, we look at how the reported differentiation results were evaluated, such as through genome or protein level expressions. Once we select a protocol to test, we then conduct the differentiation process following the published procedure. From this trial, we further evaluate the reproducibility of the result from the paper and investigate whether the expression of cell markers are observed. After we confirm reproducibility, we finally start fine-tuning the protocol within our medium. The parameters that we usually focus on are cell seeding density or total days to culture, as well as the concentrations of differentiation factors. By analyzing the culture supernatant at the same time in terms of amino acid metabolism or other amino acid related techniques, we optimize the components of differentiation medium as well. In addition to recognize the transition of differentiating cells at critical times, we meticulously track cell markers in order to obtain the appropriate cell information. So these are the example of lineage-specific cell differentiation. So in this first study, we induced mature hepatocyte as an example of endodermal lineage differentiation. In this experiment, we used 3 differentiation mediums set here. And as controls, we used B27 and KSR and FBS as a control to effectively evaluate the potential of AS401 in relation. In medium 1 and medium 3, we added 20% or 5% AS401 to basal mediums. In medium 2, although AS401 was comparable with KSR, we found that StemFit Basic 03 gave the best results, so we therefore selected it for use in medium 2. At the definitive endoderm stage, DE here, we confirmed that the expression levels of control conditions. We then continued the differentiation towards the mature hepatocyte from DE and evaluated the ALMI expression and as well as OTC expression. Both expression levels were higher in S401 differentiated cells than in the control conditions, and more mature hepatocytes were induced by AS401 than in the controls. In this second study, we induced dopaminergic neuron, DAN, as an example of ectodermal lineage differentiation. In this protocol, we mainly use 2 differentiation mediums here. In this protocol, we use KSR and B27 for control. And on day 28, cells were replated for immunocytochemistry. In both conditions, we also added 20% AS401 for the differentiation medium. So to evaluate the induced dopaminergic neurons, DAN markers such as the expression of chorin or LMX1A or tyrosine hydroxylase were periodically increased as the differentiation proceeded. As you can see, by day 28, the gene expression levels from AS401 were very similar to the controls, and also the immunocytochemistry images here, we also observed that the formation of neuronal structures and expression of tyrosine hydroxylase at the protein level. In conclusion, AS401 is a chemically defined animal origin-free differentiation media that is capable of supporting robust and consistent lineage-specific differentiation. So we believe that this novel differentiation media AS401 can help accelerate further clinical and industrial applications of promising human iPS cells-derived cell products. To this end, we hope that our CD-AOF StemFit culture systems will support and propel your research as well. So this is my final slide. Thank you very much for your attention, and I'll be glad to answer any questions.